أنا مبسوطة جدا إن أنا take part in the bioscience forum this year, and very happy to be sharing the work I've been doing for my PhD. Today I'm gonna talk about a novel approach. It's a Nobel Prize winning approach, which is a brain in a dish to study neuropsychiatric conditions. And as me being an autism researcher, obviously I'm going to be talking about autism and the autism enigma. Let me go through the contents of my presentation today and what I'm going to go through. I'm going to go through autism, the brain, how do we study and model the brain, what is the brain in a dish, and how do you use it to study the autistic brain. And then I would also want to shed light on the beauty of the autistic brain and what we can do for the autistic community. To start with autism, autism is a neurodevelopmental condition that impairs that is associated with a triad of symptoms. The first one is impaired communication. So people with autism have difficulties interpreting both verbal and non-verbal language, like gestures or tone of voice. A big percentage of people with autism are non-verbal, which makes it even harder to communicate and navigate through our lives. أول عرض احنا هنتكلم عنه هو impaired communication. communication دي can be verbal أو non-verbal. ناس كتيرة جدا عندهم توحد ما بيقدروش إن هم يتكلموا، بيكونوا non-verbal. وده comprise أكتر من 30 or 40% من people with autism, and it depends طبعا on severity بتاعته. And this is why one of the first signs of autism is impaired speech. تاني حاجة هي impaired social interaction. So even if the speech is there and even if they have verbal communication, sometimes they struggle with social interaction because they cannot read people. We have something in psychology called the theory of mind, and theory of mind is basically how you can interpret what the other person is feeling or thinking without them speaking. And people with autism have impaired theory of mind. Some other people would use the term empathy, but it's not exactly empathy. It's cognitive empathy and not affective empathy. And maybe we can talk this, and we can get this in another talk. اللي احنا بنحاول نقوله هنا ان impaired social interaction هي تاني عرض من أعراض أو تاني خصائص من الخصائص الموجودة في الأعراض بتاعة التوحد، وهي عدم القابلية إنهم يفهموا الشخص اللي قدامهم ويexpress themselves، وإن هم يقدروا يعبروا عن نفسهم بالسوشيال كونتكست اللي احنا بنعيش فيه. تالت واحد هو narrow and restricted interests. As we can see in this picture, this is all these photographs of the same boy. He's a 9-year-old boy who is diagnosed with autism, and his dad is a photographer, and he has taken these pictures just to show the severity of the condition of his kid, and trying to show the world what it's like to be in his kid's shoes. And in this picture, as we see, is like narrowed and restricted interest. People with autism do not see things as abstract. They see it as very detailed and very narrowed. They have a very restricted, we can call them obsession, but we refer from using the term obsession just because of the negative connotation that it comes with. But they have certain obsessive behaviors. When they see something, they have a great attention to detail. So if they're playing with a car, they're not going to be playing with the car, they'll be playing with the wheels of the car instead of the whole car. ده تالت عرض من أعراض الأوتيزم أو التوحد، اللي هو الفوكس بتاع الطفل بيبقى أكتر على التفاصيل الحاجة أكتر ما الحاجة كلها، وده اللي بيخليهم ساعات يبرعوا في مجالات زي الرسم والموسيقى وغيرها. طيب autism مش زي ما ناس كتيرة كانت بتكون فاكرة. autism is not a psychological condition. It's a neurodevelopmental condition, which means that it affects how the brain is developing. So autism starts at the womb. It's true that the symptoms only appear at the age of 2, but it starts at the womb. And we are trying, or the scientific community have been trying for so long to understand what type of atypical neurodevelopment is happening inside of the womb. And this takes us to the brain. So the brain is the most complex organ in the body, and for years and years and years, we've been trying to decipher this organ. We're trying to understand exactly what is this machinery and how it is working, and obviously looking at typical and atypical development in typical and atypical individuals, neurotypical and atypical people. We try to decipher how exactly the brain develops, basically. And how do we go on to study the brain? Obviously, to study the brain, we need to find the model. And that's how it works for all the organs of the body. So if we're trying to study breast cancer, we take a breast of a cancer patient, and we try like a slice of the breast, and we try to understand what exactly is going on. But since we're talking about the brain, it becomes a little bit more interesting. So to model the brain, we either can rely on animal models, which has given us a lot. The mice is one of the most prominent and most useful animal models that have been used for years, not only to study brain conditions and disorders, but also to study a lot of other, a myriad of other conditions. Mice is very close in genetically to humans, and this is why it's been really useful. But the problem with using mice to study neuropsychiatric conditions is the behavioral aspect, and how it differs to see the behavioral aspect in mice as opposed to humans. The second way to study brain is postmortem tissue, and postmortem tissue is actually known as the tissue that is taken from the body after death. So post means ba'd, mortem means wafah, and tissue means ansiga. So we're taking brain samples from people with autism after their death. Obviously, this has shown, and this has helped us a lot to unravel a lot about the condition. But sadly, because it's taken after death, it has a lot of factors affecting it throughout the span of the person's life, and obviously the availability of the tissue is also problematic. The second problem with this is that it does not show us the development of the brain. It shows the end stage of the brain. The last but not least is brain imaging, and brain imaging has been for so many years, and the advances of seeing functional brain imaging, it has given a lot to know about how the brain works, which centers of the brain light up when we do a certain task. But in our studies, or in the study of neurodevelopmental conditions, to brain imaging to learn about how the brain develops, what is atypical development of the brain of an autistic person. And obviously, because we cannot pick the brains, we cannot just like open people's brains and start looking at what goes right and what goes wrong in there. We are relying on this new breakthrough technology, which is the brain in a dish. And let me just take you through how we came across this technology and how the scientists have made this breakthrough. In the embryo, in a place called the blastocyst of the embryo, there are embryonic stem cells. Embryonic stem cells meaning that they have the properties to develop into any type of cell. So as we see here, they are within the blastocyst of the embryo, and they have the ability to form the blood cells, the cells of the nervous system, and the immune system. And how is this? Because they all contain the same genetic material of that person, and according to something called transcription factors, which we're going to call them switches, and according to each switch and where they lie, they're going to transform into the type of cell that is needed for that organ. So if we switch on the transcription factors for the circulatory system, they're going to turn into blood cells. And how did we define this? We defined them through something called the Waddington Model. That was this was the name of the mid-20th century, and that depicts normal embryonic development as a ball downhill to its final differentiated state. So let us just take a look at this, and I'm going to show you a very short video. What's at the top of the hill is called embryonic in our own terms, or for the purpose of this talk, I'm going to call them baby stem cells. And then we're going to have the differentiated state of the cells, which we're going to call differentiated adult cells. And these differentiated adult cells are the brain cells, or the muscle cells, or the nerve cells. So in this model, all the cells that are coming out of the blastocyst, or the embryo, the starting point, all look the same, all have the same genetic material. And as they go downwards in the hill, you can see the colors changing according to the switches that we talked about. We said the transcription factors are switches that make each type of cell have specific characteristics in terms of morphology, how they look, and their function. So as you can see here, they all start at the same, and then as they go down, they differentiate. So why is this important in our talk? Because I'm going to talk to you about two scientists. The first one is Gurdon. So Sir John Gurdon tried to challenge the Waddington model by trying to reverse the order of things. So in the Waddington model, we said that the baby cell transforms into a differentiated state, which is the adult cell. And this is how it goes. It goes down the hill, and it cannot go upwards. So what Gurdon did is he took an egg, so this is the baby cell, and he took a skin cell, this is the adult cell, and he tried to remove the nucleus from that egg. The nucleus is what contains the genetic material, the DNA, and he put the nucleus of the adult cell. So he took a baby cell, took out the nucleus, and then replaced it with the nucleus of an adult skin cell. Normally, what you'd expect, according to the Waddington model, that an adult cell cannot go up the hill. But what happens is that it actually goes up the hill because the nucleus of the skin cell then changes according to the switches in this egg and transforms into an embryo. So it gives baby cells. So this means that an adult cell can actually be reprogrammed to become a baby cell. So I'm just going to recap this in Arabic really quickly. الوادنجتون موديل كان بيقول لنا ان البيبي سيل ما ينفعش تقدر ان هي البيبي سيل بتتحول لديفرنشيتد ستيت ادلت سيل، بس ما بتقدرش ان هي ترجع تاني تتحول لبيبي سيل. جون جيردن ده عالم في كامبريدج قرر ان هو يتحدى الموديل ده، وخد من فروج، من ضفدعه، خد الاج بتاعتها، شال النيوكليس اللي هي بتشيل كل الدي ان اي والجينيتيك ماتيريال بتاعتها، وعمل لها ريبليسمنت، حط بدلها سكين سيل، skin يعني adult cell دي differentiated state. حط بدلها النيوكليس بتاع السكين سيل، وبدل ما يلاقي ان الاكسل دي هتتحول بالضروره ان هي تكون سكين سيل زي ما احنا حاطين الجينيتك ماتيريال بتاعه السكين سيل، لا ان هي اتحولت لامبريو. ده معناه ان هو قدر ان هو يحول سكين سيل لامبريو. ايه اهميه الكلام ده؟ الكلام ده كان في سنه 62. سنه 2006 ابتدى عالم اسمه ياماناكا في طوكيو، ابتدى ان هو يحاول يعرف ايه السويتشز اللي خلت السكين سيل دي تتحول الى بيبي سيل. So the Takahashi Yamanaka in 2006, after more than 40 years of this invention, he tried to understand what are the switches that make the skin cell transform into a baby cell. So what he did is that he took the skin cells and he started using different factors. What are these factors? These are switches that tell each of the cell to have a certain characteristic. So he started using a combination of factors. ابتدى يستخدم السويتشز اللي احنا بنتكلم عليها دي واللي احنا قلنا عليها transcription factors علشان يقدر يعرف هل هو هيقدر يريبروجرام السيلز دي من غير ما يستخدم egg of a human being or of a mice or whatever. هنا هو كان بيستخدم mice. And he has seen that he can actually, with the use of 4 specific factors, that he can actually reprogram the cells into an embryonic-like state. يعني بقت baby-like cells. طب ايه أهمية ان هو يحولها لبيبي cells؟ الأهمية في ده ان هو يقدر من البيبي cells دي يحولها لأي type of cell هو عايزه. This means that he does not need the embryo to have embryonic stem cells. He can actually get embryonic-like stem cells from adult cells. And this is what we call induced pluripotent stem cells. These induced pluripotent stem cells, or in short, we're going to call them iPS cells, are what allows us to grow a brain in a dish. Why is this important? Because you can use the cells from the patient directly to see how his brain grew, like how did it develop. So we can see the development of the brain of the patient without invading the brain, just by using skin cell. This reminds me a bit of a movie. I don't know if you'll be familiar with it. It's called The Curious Case of Benjamin Button, where Brad Pitt is brought into the world old, and as the movie goes on and as his age progresses, he transforms, like he regresses in age until he becomes a baby. And this is exactly what the Yamanaka factors are doing to adult cells, like hair, skin, blood, any adult cell. It's transforming them into baby cells, or what we call iPSC cells. So Sir John Gurdon did his experiment when in 1962 Yamanaka carried on his work. So this is on the left is Sir John Gurdon, and on the right from Tokyo is Takahashi Yamanaka, who did the experiment in mice in 2006. And in 2012, which is 50 years apart from Sir John Gurdon's initial experiment, they both shared the Nobel Prize for their collective work because they led the development of iPSCs, which is a technology that has changed the face of modeling disorders of all sorts, but especially of the brain. So although he did his experiment in 1962, he was awarded the Nobel Prize in 2012. So all the researchers up there, it took 50 years for his work to be recognized. So hang in there. So now I'm going to talk to you about like how can we use the model that I've just explained, the iPSCs, how we can use them to model the autistic brain. How can we use them in autism? So as we said, autism is a very special or a complex condition that affects all sorts. It's debilitating in all sorts of ways because it affects behavior, it affects the brain, and it comes with a lot of comorbidities. And the problem with modeling it, as I've explained, it cannot be done with mice, it cannot be done with with a postmortem tissue, and it cannot be done just by using scans. And we needed a live model. We needed a live model that shows us the development of the brain and what might be different in the development of the brain of the autistic child. So how do we use this approach? We take the samples from autistic and control patients. The WT stands for control, so wild type cells. We take the cells. It can be a hair cell, so it's completely non-invasive. It can be a blood cell. It can be a skin cell. And we use the Yamanaka factors, the switches. يبقى هنا احنا بناخد السامبلز من البيشنت. دي adult cells ما فيهاش أي خطورة، ما فيهاش أي invasion. ممكن تبقى hair، ممكن تبقى blood، ممكن تبقى skin. وبنستخدم السويتشز اللي احنا اتكلمنا عليها دي اللي هي الترانسكريبشن فاكتورز. بنستخدمها علشان نحول الادلت سيل دي اللي هي عندها خصائص الادلت سيل، نحولها لامبريونيك لايك سيل. الامبريونيك لايك سيل دي بعد كده نقدر ان احنا نحولها للتايب اوف سيل اللي احنا انترستد ان احنا نشوفه. فلو احنا عارفين ان مثلا autistic patient have certain we're interested in the glutamatergic specific type of neurons, then we get the specific type of neurons by inducing the embryonic-like cells into this specific type of neurons. And I'm gonna show you how this is done. So basically, these are the embryonic-like cells. This is day 2, and this is a time-lapse video showing how the iPSCs from the stage of the embryonic-like cells, how do they develop into neurons or brain cells that form connections and that can actually recapitulate how the brain develops. So احنا بحاول اوريكم فيديو للخلايا من اول ما بتبقى في الامبريونيك لايك ستيت، زي ما احنا قلنا من اول ما بتكون بيبي لايك سيلز، وازاي هي بتقدر ان هي تتحول لماتيور سيلز by using the switches اللي احنا اتكلمنا عليهم دول، اللي هي الترانسكريبشن فاكتورز اللازمه ليها علشان تتحول للتايب ده من الخلايا. الفيديو اللي انتوا شفتوه ده على سبان 10 ايام بس، ف in 10 days we can actually do this switch. طيب انا هنا بقى ابتدي ادخل اكلمكم على الشغل بتاعي انا بتاع الدكتوراه، وازاي انا استخدمت combination of technologies علشان اقدر اشوف ال brain development عند people with autism. So basically I was interested in a specific gene called Neuroxin 1, which is an autism candidate gene. طبعا autism has so many genes linked to it, more than 1,000, and one of them is Neuroxin 1, and it affects brain development. So I took samples from patients with autism, and I've also developed using CRISPR-Cas, and I know that the CRISPR-Cas technology, it's another Nobel Prize winning technology that has been discussed in this forum before, so I'm not going to go through it, but it's a genetic gene editing tool in which I've used it to induce the mutation of the Neuroxin 1 in my control line. And from that, I've developed more than 6 cell lines in which I'm studying how they develop, and I'm looking into 3 specific aspects: morphological, so how they look, شكل الخلايا, and by this I was doing a neurite outgrowth assay, and then molecular, so the genes, how they are expressed and what are the molecular pathways that might be involved, and then functional على مستوى الفانكشن بتاع الخلايا دي، ازاي how do they connect with each other and how do they fire, and that's how we talk when we say about neurons talking to each other, they're firing. And for this I've used calcium imaging and multi-electrode array. Very briefly, I'm going to go through some of my data, and this is unpublished data, so I cannot go more in depth with this. First, I looked at the morphology, and I tried to see whether or not the new lines might be different in neurite count, in length, and I have seen a lot of effects in that are shown in autism. So that is one very, very interesting finding, which is that the morphology of the autistic neurons were actually affected. Secondly, I looked at the molecular aspects of things. So I looked at some of the genes. ازاي الاختلاف زي ممكن يكون الفرق ما بين الحد الاوتستيك والنون اوتستيك، ممكن يكون في اختلاف ازاي التكوين الجيني بتاعه. فبصيت على ال RNA، عملت RNA seek وابتديت اشوف على الجينز اللي ممكن تكون مختلفة، وبمنتهى يعني هنختصر جدا يعني علشان اقول لكم ان زي ما انتم شايفين اي ال blue and yellow just signifies each line. So you see here the control is the first two lines, and you can see that it is very, very different from the rest of the lines. The rest of the lines are autistic-like lines. These are the mutant group. ففي جينات كتيرة جدا كانت مختلفة. بعد كده قررت ان انا ابص على الفانكشن، هل الفانكشن بتاع الخلايا دي زي الفانكشن بتاع النيورونز الطبيعية ولا لأ. وعملت حاجة اسمها كالسيوم ايميجينج، والكالسيوم ايميجينج ده بيخليني اقدر اشوف الفايرنج بتاع الخلايا، how the neurons fire and how the neurons communicate with each other through monitoring the calcium influx between cells. And basically this is one of the patients, and what we see is that they have hypersynchronicity, which means instead of the neurons firing one at a time, the whole network fires at the same time. And this is an epileptic-like function, one of the most interesting data that I have. And here's also showing the difference between the autistic and the non-autistic phenotype. طيب أنا هنا إحنا we finished the scientific part of this talk, and now we want to go into the beauty of the autistic brain. And I always like to shed a light on this because it is very important to know that not because that the brain develops in an atypical manner, that doesn't mean that it is less or that it is a disorder or a disease. It just means that it's different, and we need to learn why it's different and how it's different. And the neurodiversity that we have, and I would really like people in this talk, if they haven't heard about the term neurodiversity, to go on and read about neurodiversity. Neurodiversity allowed us to have exceptional examples and exceptional people within us, and I'm going to give 4 very brief examples because I'm watching the time. So the first is Temple Grandin. Temple Grandin at the age of 2 كان ما فيش speech, and she had severe autism, and she was asked to be homeschooled. المدرسة ما كانتش عايزة أو الحضانة وقتها ما كانتش عايزة تكمل إن هي to host Temple because of her behavior, and because they didn't see that there's any hope for her to actually learn something. Temple Grandin is now a professor at the university, so she has done her PhD, and she has a very, very successful career as a researcher in animal behavior. She's also a best-selling author, and she invented the hug box to calm those on the autism spectrum, which is also very, very impressive is that she is one of the top 100 influential people according to Times. The second example, and it's very close to my heart, is Derek Parvacini. Derek Parvacini is a music prodigy. He was born with so many problems. He is blind. He was born prematurely بعد 25 أسبوع بس. He was born with oxygen depletion that affected his developing brain, and he had severe learning disability. Derek Parvacini عنده autism. He started playing the piano at the age of 2, and at the age of 7 he was having his first concert in Tooting Leisure Centre in South London, And I would really advise you to look at Derek Pervacini, or they call him the human iPod, and see what he can do with his music. It's remarkable. It's very, very inspiring. The third example is Jacob Barnett. Jacob Barnett, until the age of 3, he was not speaking. His parents were really worried about him never developing speech, and a lot of teachers told them like he would never be able to read or write. Jacob has an IQ that is higher than Einstein, and at the age of 14, he had already started his master's degree. So he finished his undergraduate by the age of 14, and he was tipped for a Nobel Prize by the age of 14 or 15, I don't remember. The fourth example, and the closest to my heart, is Karim al-Wazin. Karim is an Egyptian Italian autistic adult. Now he must be 26 or 27. I have seen him when he was 22, and he has held a beautiful gallery inside Sawi for those who Egyptians watching us. He was extremely talented, and what he did in this exhibition, he painted all his feelings, how he sees things, and he made this incredible exhibition. I've had his paintings for years now, and I love how they depict exactly his feelings and how he sees the world. So what you can do for the autistic community: awareness is key. You need to learn about how these people perceive the world and try to welcome them into our world. You need to do more to make them feel more accepted, and we need to include them by making the environment more welcoming to them. So now there's a lot of things happening in Egypt to help with the inclusion of autistic people in our community. So, for example, we take care of the lights and the noise because of those with hypersensitivity or sensory overloads. And finally, end the stigma. And I would really, really advocate for ending the stigma by ending the use of certain words like that we generalize and use in our daily lives. When we say mental, psycho, OCD, depressed, panic attack, bipolar, please do not use those terms unless there's a diagnosis, because what you're not helping doing is raise awareness to these terms. You're trying to generalize them and make them less serious than they are. You would never say that about a cancer patient, so please be cautious of that whenever you can. And of course, every 2nd of April, and since we are not so far from that, is Autism Awareness Day, and April in general is Autism Awareness Month. And as part of the autism awareness campaigns, every monument, every landmark in every country lights up in blue to raise awareness for the condition. And lastly, I would just like to thank you and thank my team that allowed me to do the work that I'm doing in my PhD and my postdoc as well. And thank you for listening.